All right, well, welcome everybody. We'll go ahead and get started here. I'm super excited that you're all here. I want to thank you all for being here today. Today we have some exciting stuff to talk about. We're going to be talking about LAMP and its role in the point of need detection, as well as giving you some solutions to some common problems that researchers face in this area. Before we get to the more exciting topics, a little bit of housekeeping on my part. My name is Anthony Austin. I am the Global Marketing Manager here at Medix Biochemica. And first off, you'll notice that there is a Q&A button at the bottom of your screen. Please feel free to use it to submit any questions you have now or during the talk. We will hold a session at the end of the webinar where we'll go through all the questions that we can. However, with the time being limited, we may run into a time issue where we can't get to all the questions. However, rest assured that we will follow up with you with an email after this to get to all the questions that we discussed and the ones that we weren't able to get to. And if you're anything like me, the questions don't even come until later when you're taking a walk or you're driving home from work. So if that is the case, please feel free to reach out to us at info@medixbiochemica.com. That's info@medixbiochemica.com. And give us any feedback, questions, comments, or discuss any interesting opportunities that you might see with us in the future. This webinar is also being recorded, and we will also follow up with an on-demand link so that you can see the slides in the future and review the recording as well, or even share it with colleagues as you see fit. A little bit of background on who we are. We are Medix Biochemica. We're an independent, industry-leading, and international raw material supplier. We specialize in providing high-quality, high-performing antibodies, antigens, biospecimens, proteins, enzymes, molecular diagnostic reagents. We like to say that if there's any critical raw material you need, chances are we can provide that for you. We do have lots of expertise in immunoassays, as well as clinical chemistry assays, molecular diagnostic reagents, and a number of different similar fields in that area. And in fact, our molecular diagnostic manufacturing site is located in the beautiful city of Constance, Germany, where we specialize in engineered and unique polymerases for DNA and RNA. We are ISO 13485 certified, and we have lots of in-house expertise and experience, especially in the areas of assay development services, as well as lyophilization services and products. And today, I'm excited that we're joined by two scientists from the Italian Institute of Technology. First off, I will hand over to Paola Cecere, who will walk through the presentation for today. And then in the Q&A session that I mentioned earlier, we're joined by Pier Paolo Pompa and our very own Giuseppina Stanino, who will walk you through some of the questions that you have. And without further ado, I hand over to you, Paula. I will stop sharing my screen.
Great. Okay, thank you, Anthony. Welcome to everybody and good afternoon. I'm Paola Cecere. I'm a postdoctoral researcher at the Italian Institute of Technology. In particular, sorry, it doesn't work. Okay. In particular, I work in the nanobiointeraction and nanodiagnostic group, where we develop integrated smart and rapid assay sensor for point-of-care diagnostic, using different strategies, including nanomaterials, nanozyme, surface biochemistry, and molecular biology. In this context, one of our applications is the development of LAMP-based genetic tests. LAMP is the short name for loop-mediated isothermal amplification, an outstanding reaction for DNA and RNA amplification with broad and multifaceted capabilities. Tau LAMP is known for being robust and accessible, and mastering design is required to maximize its elevated performances, bringing out its full potential. So, driven by this goal, IIT and Medix Biochemica merged their expertise to create our LyoLamp, a diagnostic platform prototype for genetic testing. Here's the agenda that we planned for you. The webinar will be divided into two sections. In section 1, we will learn something about LAMP, how LAMP works, its strengths and challenges, and where LAMP can be carried out. In section 2, we will introduce you to our LyoLamp, discovering its identity card, some in-depth aspects, and some of its applications. So, how does LAMP work? The answer is in the name, loop-mediated isothermal amplification. Isothermal means that, unlike PCR that uses thermal cycles, LAMP work at a single reaction temperature between 60 and 65 Celsius degree without a denaturation step. The reaction is mediated by loop, indicating the loop stem structures, where there is a single-strand region, the loop, and a double-strand one, the stem. Before we dive into the LAMP mechanism, I'd like to share with you some of key points that we have to keep in mind. First of all, LAMP uses a set of not two, but from 4 to 6 specially designed primers, specific for from 6 to 8 target regions. The first pair of primer include the inner primers, FIP and BIP. These primers start the reaction. As you can see, they have a unique shape with two portions. One, F2, is complementary to a portion on the DNA target, and the other, F1, is identical to another region on the DNA target. This particular shape helps in forming loop structure during the amplification. The second pair of primer includes the outer primers F3 and B3 that are complementary to the outermost regions on the DNA target. The last pair are the loop primers that are optional primers, and they work only in stage 2, acting as accelerating primers. The second key point to understand is that LAMP bypasses the PCR denaturation step through 3 mechanisms. At 60-65 Celsius degrees, the DNA target maintains a dynamic equilibrium, meaning that an amount of the DNA target is in the single-strand form, and so it is already available for the amplification. The LAMP works with a DNA polymerase with a high strand displacement activity, meaning that the polymerase synthesizes the new strand while displacing the existing one. 3, each stem loop primer has 2 priming sites. One where the primer anneal to the single-strand loop, and the other where the self-priming occurs at the free prime end of the stem. So, LAMP uses 2 stages. In stage 1, there is the production of the starting material for the stage 2, where the exponential amplification takes place. At the reaction temperature, the DNA target is partially denaturated. This means that the inner primer FIP can anneal to the single-strand form of the DNA target. So the primer is elongated by the polymerase. On this same strand, the outer primer F3 can anneal. In this case, the polymerase elongated the F3 primer, displacing the FIP elongating strand. and releasing the first single-strand DNA, so the first product of our reaction. In this strand, thanks the particular shape of the FIP primer allows the self-hybridization between F1 and F1C, creating the first stem loop structure. The same mechanism that we have seen till now is repeated. So on this strand, on the opposite end by the backward primers. So we have the inner primer B annealing and its elongation, the outer primer B3 annealing and its elongation, and the strain displacement of the BIP elongated strand with the self-hybridization of the complementary regions, releasing this particular structure called dumbbell-like structure that is a single-strand DNA with a loop structure at both ends. This dumbbell-like structure is the starting material for our step 2. It is an artificial single-strand DNA target. So, on this structure, we can have 3 amplification starting points. The first is the annealing of the inner primer to the loop. The second is the self-priming at the 3' stem end. And the third is the annealing of our accelerating primer that work only in this stage to the other loop. So, combining multiple amplification sites with loop unfolding and refolding strain displacement activity, the exponential amplification in this stage generates a very high concentration of very complex amplicon, as you can see in this picture, with different sides, the so-called cauliflower-like structures. So let's move on to our second question. What are LAMP strengths? The PCR is recognized as the gold standard technique for molecular biology. But LAMP offers a series of advantages that make it a speedy and hassle-free tool for on-the-spot diagnostics. Thanks to its isothermal mechanism, LAMP uses a simpler instrumentation than the complex thermal cycle required by the PCR, cutting costs and making LAMP a more affordable choice. Moreover, LAMP is more tolerant to inhibitors than PCR, allowing it to work with crude sample and simplified extraction protocol. Additionally, LAMP has a higher amplification efficiency and so a greater air yield from the same amount of starting material. But what about speed, sensitivity, and specificity? It is a common belief that LAMP, while faster, is less sensitive and less specific than PCR. It is not truly entirely true because these 3 parameters are highly dependent on the LAMP design. So a high-quality design can result in a high-performing LAMP. So in a high-performing LAMP, the reaction can be completed in just 7 to 40 minutes, shortening the turnaround time if compared with PCR that requires several hours. In a high-performing LAMP, we can have a greater sensitivity as low, detecting as few as from 1 to 3 copies per reaction, compared to the 10 to 100 copies in PCR. Moreover, we can have a greater specificity by targeting 6 to 8 template regions instead of the 2 target regions in PCR. So, as you can imagine, designing such an advanced assay can be very complex. So now let's consider the other side of the coin. Which challenges do we face with high-performing lamp? First of all, the primer design that is very complex and that can impact the overall assay performance in terms of sensitivity, specificity, reaction rate, reliability, and reproducibility. Moreover, the primer design can be very tricky in detecting challenging DNA markers as SNPs. The complexity lies in the design of not 2, but from 4 to 6 primer that must meet specific chemical and thermodynamic requirements. This primer must ensure a specific detection with an undetectable background while avoiding non-specific binding and primer damage. So an extensive expertise in primer design is required for high-quality primers. The second challenge is the optimization of the mix composition. That is the second factor that can affect the overall assay performance. LAMP requires the fine-tuning of every component concentration, utilizing high-efficient warm start polymerase and lyo-compatible reagents. The third challenge is the high risk for carryover contamination, a well-known drawback in lamp-based assay, and that is due to the high concentration of amplicon. So for this reason, the assay must include a system that prevents cross-contamination. Lastly, the extraction protocol used in a lamp-based assay must use lamp-compatible reagents. So, where can LAMP be carried out? A high-performing LAMP can be particularly suitable for point-of-care and high-throughput genetic testing. LAMP is ideal for point-of-care, giving rapid results at the point of need, where we need a fast turnaround time, user-friendliness, affordability, and portability. It is a perfect tool also in high-throughput testing for continuous and large-scale screening, where scalability, high sample capacity, automation capability, and reproducibility are required. So, in summary, the main requirements for POC and high-throughput testing design are chemistry, workflow, supply chain, and instrumentation. Captivated by the great technology, the great potential of this technology, IIT and Medix Biochemica merged their expertise to create LyoLamp, our diagnostic platform for genetic testing that combines a high-performing lamp with the convenience of the lyophilized format. Every detail of LyoLamp has been developed to be smart, user-friendly, and business-friendly. Smart because LyoLamp offers all-inclusive solution that include DNA extraction and the amplification step. It combines a high-performing lamp where the lamp primer set is included and a lyophilized format. User-friendly because the LyoLamp is an highly tolerable test featuring non-invasive sampling for clinical application. It is easy to use and it requires a minimal instrumentation, enhance its portability for point-of-care applications. It is business-friendly because it is cost-effective and it offers alternative solutions to traditional methods that not always are already available on the market. Its scalability and the possibility to develop custom on-demand solution make LyoLamp an attractive option for various market needs. Thanks to our extensive expertise in primer design, LyoLamp is suitable for the detection of different DNA markers, such as species-specific genes, SNP genotyping, and indel genotyping, for different targets from human and bat genome to microorganism and food. This flexibility makes LyoLamp suitable for different applications, from infectious diseases to genetic predisposition and susceptibility, pharmacogenetics and nutrigenetics, food safety and traceability, environmental monitoring, and bioterrorism. LyoLamp uses 3 simple POC compliance steps. In the first step, we have the sample collection. We can test different types of target, human, vet, food. For human and veterinarian application, we use non-invasive samples as saliva swab and fingerprint. The second step uses a streamlined protocol for DNA extraction exploiting a thermochemical method. A ready-to-use extraction solution with a room temperature storage is already provided with the test. The DNA extraction requires only a simple heater and a few simple operating steps. Importantly, LyoLamp doesn't require a DNA purification, avoiding this complex and time-consuming step. The total extraction time is around 5 minutes to 10 minutes only for some application. The third step is our LAMP amplification step, with a total reaction time from 7 to 40 minutes. This step combines the high-performing LAMP with a specific primer already provided and the lyophilized technology with a real-time fluorescence-based detection. Our assays are open test without an associated device. They require a simple and widely available instrumentation. The test design can prevent the LAMP carryover cross-contamination, both using a one-pot closed-tube format for amplification and detection, and integrating in the LAMP step an enzymatic system for the contamination degradation. LyoLamp uses a customized lyobead technology, where LAMP mix is pre-dispensed in freeze-dried spheres, with one bead corresponding to one reaction. This technology has been developed for a simple handling, that requires that the lyobead is resuspended with that buffer. And then we have the addition of the DNA extract. Our lyobeads are ready to use, and their composition has been optimized for our applications. This technology ensures a long-term stability with a room temperature shipping and storage. Importantly, there is the possibility to scale up the production, for example, in bulk for high-throughput testing. In these plots, we compare our lyobeads with the reagent of one of our main competitors in solution with the same primer set for the detection of Salmonella enterica. As you can see, LyoLamp reaches the same sensitivity with faster reactions. So now let's explore some LyoLamp applications, starting from the real-time SNP and indel genotyping. SNP, single nucleotide polymorphism, are mutation of a single nucleotide, while indels, insertion-deletion polymorphism, are mutation as insertion or deletion of more than one nucleotide. SNPs are a median market because they can unlock very crucial information. Indeed, they are involved in development and progression of different diseases, in genetic susceptibility and predisposition to specific conditions, in pharmacogenetics, and so on. On the other side, they are a challenging marker because SNP genotyping requires a complex and expensive procedure that often show a high background signal and require a high quality DNA. Traditional methods based on microarrays, PCR, and sequencing are not for their feature are not suitable for point-of-care testing. So now we will understand the detection strategy of our lyolamp in SNP genotyping. It is important to understand how our simple and user-friendly technology can discriminate a single nucleotide. In SNP genotyping, lyolamp uses a double tube solution, where each tube contains a probe that is specifically designed to target either the mutated or the wild-type SNP. That is to say, in tube 1, we have the MUT probe specific for the mutated SNP, and in tube 2, we have the wild-type probe specific for the wild-type SNP. Each probe will have a perfect match with the specific genotype and a single base mismatch corresponding to the SNP with the other genotype. For example, MUT probe with wild-type SNP genotype. The heterozygous genotype will show a perfect match with both the probes. Lyolamp for SNP genotyping is designed so that if there is the perfect match between probe and genotype, the lamp reaction is fast. If there is the single base mismatch, the lamp reaction is slower. This different speed is visible in the amplification plot over time, where the amplification curves related to the slow reaction will shift to the right, to longer time, okay? So in this plot, the blue curve is the fast lamp at lower time, because thanks to the perfect match, it requires less time to achieve the amplification. The red curve is the slow lamp. Lyolamp uses this time gap for the test reading and the single base discrimination. In the assay, we add the extracted DNA from our sample, unknown sample, to both the tubes. So each sample is tested with the two reactions performed in parallel. So for each sample, we will have two amplification curves, one for the fast reaction and the other for the slow reaction. The unknown SNP genotype is detected simply by identifying which tube corresponds to the fast reaction in the amplification plot. It seems so complex, but it is very simple. If the fast reaction corresponds to the MUT probe, the unknown SNP genotype will be mutated homozygous. If both the tube will give a fast reaction with the overlapping of the amplification curves, the test result will be heterozygous. If the fast reaction will correspond to the wild-type probe, the test result will be wild-type homozygous. So to sum up, how we can use lyolamp for SNP genotyping is the illustrative workflow. As mentioned, lyolamp is an all-inclusive test. So it provides for each sample a ready-to-use extraction solution and two tubes, one with the mutated lyo bit and the other with the wild-type one. The test uses three simple steps. the collection of a non-invasive sample, the 5-minute extraction without the purification step, and the amplification with the lamp reaction. In the first step, in the amplification step, we just add the DNA extract into the 2 tubes with the lyo beads. The detection is based on the fluorescence signal, simply reading the time gap between fast and slow reaction. The test is completed within 30-35 minutes. So with this solution, we have a molecular test for SNP genotyping that is completed within 35-40 minutes, including the extraction step. This table lists our lyo-lamp test for SNP and indel genotyping. We have already developed this test, but lyo-lamp is customizable for on-demand targets. So these are only examples. In this example, you can see that we applied lyo-lamp for the SNP genotyping in the case of the genetic predisposition to lactose intolerance, where, as you can see, the fast lamp is for lactose intolerance, the mutated reaction, and for lactose tolerance, the wild type 1. So as you can see, we have a very, very simple test readout. Lyo-lamp, the use of lyo-lamp for SNP genotyping can be extended also to other fields, for example, for food fraud and traceability. For this application, we collaborated with Barilla, a leading company in pasta production that produces pasta using only a unique variety of wheat. So, in this case, we use our lyo-lamp for SNP genotyping to develop a test for the varietal discrimination. This kind of test can be useful for industrial quality controls to check, for example, the purity of raw materials and to identify adulterated products. As you can see, in this case, we successfully identify even low percentage of adulteration. The third macro area for lyo-lamp application is the microorganism detection. In SNP genotyping, we adapt our lyo-lamp for the discrimination of a single nucleotide. For microorganism detection, we made lyo-lamp ultra-sensitive. Indeed, detecting the lowest possible number of target copy number is crucial for this application, both for the clinical practice, for the early diagnosis in clinical practice, and for routine quality control to ensure compliance with the regulatory standards. As you can see, lyo-lamp for the microorganism detection is is very sensitive, detecting as few as 2 to 10 copies per reaction within 10 minutes or less, to up to 20 minutes only for some applications. In this table, we provide an overview of our lyo-lamp test for microorganism detection that we have already developed. But also in this case, there is the opportunity to customize our lyo-lamp for on-demand target. This is a practical application, the diagnosis of Mycobacterium tuberculosis. Traditional methods can be quite quite complex, but there is a point-of-care test available on the market that uses a PCR method. It requires 77 minutes and it uses a specific device. So it seems less advantageous if compared with our Lyo lamp, where we are ultra sensitive. We use an open test, so without an associated device, and we have very fast reaction in within 10 minutes. Here there is the illustrative workflow for the detection with Lyo lamp of infectious disease. Also in this case, we have all-inclusive test with the ready-to-use extraction solution and a single tube in this case for the amplification. Also in this case, we have 3 simple steps. The sample collection with an non-invasive procedure, the 5-minute extraction without the purification, and the amplification step where the DNA extract is added to the tube. These tests are on-off tests. Okay, so the detection is based on presence or absence of the amplification curve. And they are completed within, they are very fast. They are completed within 10 minutes, up to 20 minutes only for some applications. So in this case, we have a molecular test for the microorganism detection completed overall within 10 to 15 to 25 minutes, including the extraction step. So in conclusion, our Lyo lamp is an all-inclusive diagnostic platform that combines a high-performing lamp and a lyophilized format. Thanks to its smart design, it is fast, sensitive, and specific, and it doesn't require a DNA purification. All its features make Lyo lamp user-friendly and business-friendly, and suitable for point-of-care and high-throughput applications. The technology is fully customizable. We can test different types of samples and different volume of samples. Moreover, the technology flexibility allows to detect different DNA markers as challenging targets as SNPs and ultrasensitive detection as a microorganism application. Its versatility makes Lyo lamp suitable for different applications, spanning from clinical practice to food safety and traceability and environmental monitoring. So that's all. If you are interested in collaborating, we will be excited to hear from you. Thank you.
Thanks, Paola, and I will share my screen with just a quick couple slides. Let's see. There you go. First off, a massive thank you to everybody to take their time to be with us today, and especially thank you to you, Paola, for taking us through there. If you enjoyed the webinar, or maybe even if you didn't, please use this QR code, and I will actually send a URL link that's clickable and a little bit more user-friendly in the chat here in just a minute. But we want to make these webinars exciting, interesting for you, so please tell us how we might be able to improve there. And if you enjoyed it and wanted to hear about the next event that we have, please also use this QR code to just subscribe, and so that whenever we send out the notifications, you'll be sure to be on the list. So with that, and just by the way, all those QR codes that are only on the screen for just a minute, this will be in the slides that we follow up with you on. So apologize for that going out of the screen so quickly. Now for the Q&A, I will turn it over and invite our panelists back, and let's go through some. First up, to Paula. How does the operator's ability to interpret Lyo lamp to interpret lyolamp results impact the overall effectiveness of the test?
Okay, lyolamp has been designed to be user-friendly in every aspect and in every step, including the test readout. Okay, so thanks to the test design, the test readout is made as simple as possible. For example, in the microorganism detection, we have an on-off test where we have to ask, is there the amplification curve? If yes, the test is positive. If no, the test is negative. So we have a very simple readout. Also for the genotype, we have a simple readout requiring only the reading of the time gap between fast and the slow reaction. So in this way, thanks to the design, the test design, even a user with a low technical expertise can run the test and read the result.
Okay. And I will go through this next one. Thanks again for everybody for submitting the questions. So next one we have is, after COVID-19, the use of LAMP has increased, but why are there not many commercial kits available?
Okay. Yeah, there are no so many kits available on the market. Maybe because integrated lyolamp in routine practice is very complex. I think for two reasons. First of all, designing a lamp-based assay that can truly compete with the PCR is very challenging. The second reason is that medical, scientific, and maybe also industrial settings still don't accept an alternative to the PCR the PCR technology, despite the advantages of LAMP for some application. Moreover, there is the common belief that LAMP is less sensitive and less specific, and also that one of the main drawback of LAMP-based assay, the cross-contamination, are out of our control. We can control them, but as we can say that this is not true.
Yeah, I would like just to add something, because I think that it is important to remark that, as Paola showed in the seminar, it is not just a matter of designing good primers, because yes, you can have a very rapid reaction, but the point is to take care of all the steps, making the assay user-friendly and very clear to the user. So the point, as Paola mentioned, was that it is important to get to have very good extraction kits, very simple. We have to avoid the purification step, and we have to take care of all the chemistry and primer design. I just want to remark that if one just look in the scientific literature, you can find a very recent paper showing lamp amplification that takes 1 hour, and they are run after very long purification steps, so that your lamp reaction finally takes 3 hours. So of course, one cannot see any advantage with respect to a very standardized method such as PCR. So instead, on the other side, if we can take care of all the steps and design a very affordable reaction, you can have let's say a very good alternative in which in 20-30 minutes you can run your assay in a very simple way.
Excellent, thank you. I know we're running up a little short on time, but I'll and actually a couple of the questions that were asked kind of go along the same lines as that one. We'll go through a couple more here. Next one we have is, can the lyolamp beads be applied to any other formats? For instance, 96 well plate format.
Maybe I can take this one. So yeah, from our experience at Medix, we can produce in general lyobits in different sites, and therefore we can apply also the lyolamp prototype as any other lyobits in a different format, including also a 96 well plate. For example, we already have some products out of shelf where we have lyobits that contains standard qPCR master mixes, and these plates are breakaway plates, so you don't need to run 96 samples at the same time, but you can break them and just run the samples that you need. However, we can also adjust the size of the beads to fit them in a single tube, or sometimes in a point of care devices you have some microchips, so we can also adjust the size for that. And the lyolamp prototype is one example of this.
Excellent, and it's quite versatile, and for the sustainability conscious out there too, with those breakaway plates are quite useful.
Yeah.
Yeah,
our customer appreciate this because many are afraid to purchase there because not all the time you have the need to run 96 samples at the same time. So this really helps. Yeah. Also, as you said, the sustainability, but it's also cost effective.
Yeah, for sure. I think that speaks to all the lab scientists on the call. Well, let's go through two more here, and then we'll wrap up for the day. So the next one says, to what extent can the colorimetric detection be effectively utilized in this application, and what are the benefits and limitations associated with that?
Okay, I can take this one. We have already developed a lamp-based assay using colorimetric detection, where the readout is based on a color change that is visible by the naked eye. This kind of detection has some advantages and some disadvantages. For example, it requires a far simpler instrumentation that if compared with a lamp with a fluorescence-based detection, because we can use a simple heater since we have a naked eye readout. So it can be suitable for example, low resource settings. On the other hand, a colorimetric detection requires a higher turnaround time, and moreover, it requires the fine optimization of the color change, because this color change must be objective, user independent. Okay.
Yeah, but anyway, Paola, we can add that in case it is required, we know very well how to run also this kind of assay. They take us just some longer time, but they are very efficient, and they of course also in this case you can reach single molecule sensitivity and just by inspecting by naked eye. This is an alternative.
Excellent. Thank you very much. So I'll wrap up with one last question. There's a couple questions about the stability and resistance to humidity. So how stable and resistant is lyolamp to humidity?
Yeah, so as I mentioned before, and also Paola during the webinar, so the lyolamp is still a prototype, so we have not fully assessed the stability for this specific prototype. However, also Paola and Pierpaolo ran some initial stability test in their laboratory, and could find that so far they are quite stable. However, what I can say in general for the lyobits that we produce at Medix, we can ensure a stability up to 2 years if they are sealed in a proper way. And also another things we are working on, because we know when then the user gets the beads, especially in a bulk format, and they need to move it in a a 96 well plate or a chip or whatever, they need to work in a humidity controlled environment, and this is not always possible. Therefore, we are trying to develop a new protocol to have lyo beads that are humidity resistant, and this way this will simplify the workflow for the user and also be more cost effective. And as I mentioned, for lyolamp, we are still assessing it, but we expect to reach more or less similar stability once the whole experiments are ended. I don't know if Piero or Paola want to.
Yeah, I just can confirm what you are saying. We found the lyo beads extremely, let's say, easy to use. Of course, we don't have long-term stability testing, but our direct experience is that we leave these beads, let's say, 6 or 8 month old somewhere in the lab in ambient conditions, and they work perfectly after 8 months. So I don't see any kind of problem, because we even open them and just leave somewhere. Of course, it is not an high humidity environment, but I think it is very promising. Yeah.
Excellent. A real life stability, as they say, real-time stability.
Yeah.
All right. Well, I know we're close on time. I will just bring up, since I completely fumbled it earlier, I'll bring up the QR code so that you can subscribe to our webinars in the future. A massive thank you to everybody who was involved behind the scenes and on the scenes. Thank you so much for taking us through this. Again, if you have a chance to click on that feedback survey, we want to improve for you so we can make these webinars as relevant for you as they can be. If you any questions come up, please reach out to us at info@medixbiochemica.com. And if your questions are specifically for our Italian Institute of Technology colleagues, we'll be sure to get in contact with them as well. And if nothing else, enjoy the rest of your day, and thank you again. Bye, everyone.
Thank you.
Thank
Bye.
you. Bye-bye.